James Gunton from Lehigh University in the US. Professor Gunton got his degree in physics in Oxford University and then he did a PhD at Stanford. Also, just learned that he has a degree in chemistry. Probably also
I never tell anyone that.
Some other people also have those degrees.
He knows.
Okay, then after this stay in Australia, he came back to the US to Temple University, established himself as one of the leader troops in the university for 20 years. Then he tried administration for a while. He went to as a provost to Kenyon College in Ohio. He didn't like it much, or at least not enough, so he went back to, he never quit fix actually, but he really did administration kind of. He was doing a little of research. And then he moved to Lehigh University. That was in 1994. 1988, sorry, 1988. So he's spent a couple of years on administration. He's been a professor in physics in Lehigh University for that time. And now, just to show you how respectable he is, he has a chair with a name. It's the Joseph Walsmith Chair of Physics at Lehigh University. So not everybody has a chair with the name of somebody. Okay, he's been a visitor of many universities, just to mention a few in Japan, in Kyoto, Fukuoka, in Switzerland, Geneva, Lausanne, Institute for Japan in France, and then in Zurich in Germany. He's been a regular visitor to our institute, previously in Lehigh, the physics department, now it is for many times more than I can remember. His work is very broad. His earlier work included critical phenomena. He was at the time with exponent inequality, my social group, scale that was coming up. There is even an exponent inequality that bears his name, the Gunton-Buckingham inequality of critical exponents. Also over Bose-Einstein synchronization, the normalization group in equilibrium, and then later in dynamics. He did very interesting work with Kawasaki on mold coupling theory. Then kinetics of phase transitions, which is himself as one of the world leaders, and the reference in the work that he did in the 70s, 80s in Temple University. In charge transport semiconductors, pattern formation, polymer dynamics, spatiotemporal chaos, protein condensation, and many others. He has written many papers, but also review articles. I would like to mention one of them in collaboration with Maxi on the kinetics of first order phase transition. We used to call that the Bible. We used to know how. And as a Bible, it also had a small New Testament, which was the book in Michel Ross on nucleation theory. That was small. So it was a big book and then the smaller book on the phase transition and nucleation theory. Currently, he's doing research on protein condensation and protein crystallization. And it's therefore of interest first to determine the properties of the protein themselves and also relevant in certain human diseases as Sickle will tell us today. Well, there are many good reasons to have Professor Gunton here with us today. He's been a long time collaborator with us. Both Max and I postdoc with him while he was in Temple. He had a large group that he leaded, a group that spanned over many activities, not just kinetics of phase transitions. From him we learned, besides physics, we learned many things, mostly about the attitude towards science. Science has to be done with devotion, with rigor, and with hard work. He's a hard worker. He was here the last week. Okay. He has strong leadership capabilities. He had one of the largest groups on kinetic phase transition at the time at Temple University. And he still managed to help that group together with very good friendship and with excellent results. Besides that, we should treat him very well because he's a member of our advisory committee. Also give him a good impression so he can right now produce a lot of
I'm really embarrassed. And I feel I should issue a rebuttal. I've been coming here since my old son Tomas was born. That's quite a while ago. Speaking of the Bible, in the 1980s, I was traveling to Switzerland and I was going to give a talk on mode coupling theory. and I was reading a famous review article by Kawasaki. And an African-American man came into the car I was sitting in and asked what I was doing, and I said, well, I'm reading the Bible. At which point he gave me a lecture on I should convert myself to Christianity. Well, I'm a little sensitive to that. Okay, today I want to give you a somewhat broad overview, including some work we've done on self-assembly of proteins from aqueous solutions. Oops, I have this problem with this. I want to start by emphasizing that this is not really limited to proteins. There's a lot of work going on now in the States in colloidal science, which is probably a much better funded area of research than proteins, where you have very similar phenomena. And I'll try to give you an example of that. One deals a lot with the kinetics of the clusters that form under certain conditions from either proteins or colloidal particles. And then I'll talk in particular about some work we've done on the kinetics of microsphere formation in a certain model of insulin molecules in solution. And then I'll talk about some work we're doing right now that has relevance to sickle cell anemia, which has to do with the formation of bundles inside red blood cells that lead to the disease. And if I have time, I'll briefly mention some work that Tony Perez, who comes from here, has started with our group. So let me start first of all with proteins.
You should stand there.
Stand here? Oh, sorry.
No, because of the
Oh. This is another thing. You can see I'm no longer the boss. I started in this field a few years ago because I found out that scientists were trying to understand theoretically how to grow high-quality proteins from solution, high-quality protein crystals from solution. And that was of interest to the biologists who want to determine protein structure. And so crystallographers were trying to grow high-quality protein crystals in order to give them to X-ray diffraction people who would then determine the structure. Turns out that's very difficult to do, and it's still done in somewhat of a hit-and-miss process, even though there's been some theoretical progress in understanding this. My real interest lately has turned to this issue that some diseases are caused by undesirable aggregation or self-assembly of proteins from solution. These include cataracts, sickle cell anemia, and Alzheimer's disease. And there the goal is to try to figure out, to understand the process of the self-assembly in order to be able to change the conditions in such a way that you can actually slow down the phenomena or prevent the phenomena. And then there are examples in drug delivery, including the one I'm going to talk about now, where you need to have, for example, if you're going to get an injection of insulin, if you have diabetes, for example, people want to have a relatively monodisperse distribution of insulin microcrystals in solution in order to have sustained drug delivery so that you don't have to have too many injections over a period of time. Now, it turns out that there are several problems in colloidal self-assembly that are very similar to the problems in protein aggregation, and there really has been a lot of give and take between the models one uses between the two fields. For example, the classic work in proteins that got me interested was actually done by my president of my university, who's a chemical engineer, and she discovered something quite interesting in colloidal science about 20 years ago. Nowadays, some of the models that people such as myself are using in the protein field are being used in the colloidal field. So this is a very rich interplay. Colloidal scientists are in principle trying to design smart materials. That is to say that you try to prepare a colloidal particle, for example, with interactions between each other in such a way that when you put them together, they will assemble into a desired structure or superstructure. For example, a diamond lattice for a photonic crystal or something of that sort. Very recent work, which I'll give you an example of on Janus or Janus particles, I'm not quite sure how you pronounce that, where you actually can pair a colloidal particle so that part of it is hydrophobic, for example, or part of it is, the other part is charged, and you can control the nature of the interactions between these colloidal particles. And because one hemisphere might be charged and the other hemisphere not, you actually can have directional dependent interactions. That is, that the interaction between two particles depends on their relative orientation. That makes the modeling more difficult, but also the physics more interesting. And that's the last topic. That's actually what Tony Perez is working on with me, which is a big pain. So let me just first of all give you some examples of self-assembly. Talk about human cataracts in a second. Amyloid clusters, which there's been a lot of work done on. Dental microribbons in the formation of enamel and teeth. Janus particles, and then very briefly protein crystals. I'm not really going to talk much about the crystal part today because I've talked about that before here. So there are different types of cataracts, but the type that I've been working on has to do with, you see them here in this illuminated picture. What happens in the human eye is that you have a family of proteins called crystallins, and there are three types, alpha, beta, and gamma. The important class are gamma proteins because they can actually exert weak attractions among themselves, and if the physiological conditions are such in the eye, those gamma crystallins can actually aggregate together and form little clumps that then scatter the light, and that is what causes your formation of cataracts. George Benedict at MIT, who was a famous physicist when I was a graduate student, has done some of the classic work on this subject and found out that there's a phase transition that takes place, in vitro at least, where you have a fluid-fluid phase separation where some of the gamma crystallins go into a more dense phase, and you get little droplets of these gamma crystallins in dense phases. And that, as soon as you get that, that's the thing that triggers the formation of these little clumps areas. And he actually formed a company years ago where his idea was to lower the critical point at which this phase separation took place so that it was below the physiological temperature at which our eye operates. And actually he had great success in the scientific part of it because he was able to have, he found eye drops that you could put into rats and things of this sort that would lower the temperature so that you were nowhere near the phase separation point, and that prevented this from happening. The problem was when he got into stage 3 with the FDA, it turned out that the eye drops, when you put them into humans, caused more trouble than the cataracts. So that was the end of his company. But this is now, it's still going on. I just refereed a paper by him in which he points out that the beta crystallins, which normally are ignored because they don't have attractive interactions, actually, just by their presence, have a weak interaction with the gamma crystallins and actually suppress the phase transition. So people are going to have to start modeling that to get further understanding. Now, I'm not an expert in Alzheimer's disease, but it is thought that the plaques that form that are the cause of Alzheimer's disease come about from the aggregation of certain types of proteins into what are called these fibrils. And there's a scientist at the University of South Florida, Martin Muschel, who did some of the classic work on lysozyme, which is a paradigm of protein crystallization. He has turned to this subject and trying to understand how you actually get these fibrils that are known to form that then lead to these plaques. And this is actually a study of his which shows, I think you can see it here, that you first form from the monomers, in this case lysozyme, which he's using as a test case, you form little oligomers. Then the oligomers form dimers of oligomers, and then these form together into these long chains. And then these chains continue to form these fibrils. That's something that we're actually now starting to model to try to understand the kinetics of how this process happens. It all is due to weak attractive forces that result from changing of the conditions, in this case of the lysozyme solution. So that's another example of the kinetics of cluster formation where you really have to understand the details to make progress. This is something that we're actually trying to model right now. Tony, in fact, is starting on this. It is known that, I don't know this, I don't want to, others know, that this protein, amelogenin, which is about 90% of the process of controlling the formation of enamel in the tooth, actually does that in part by self-assembling into nanospheres, which I'm going to show you a cartoon of in a second. Those nanospheres then coalesce together to form these micro ribbons that you see here in this real picture, where there's been a detailed study of the kinetics of this process. So the idea is, and this is what they see more or less when they look under the microscope or do light scattering, is that you start with this melogenin, which is primarily a hydrophobic sphere with a hydrophilic tail, which consists of several amino acid residue groups, and that is weakly charged. And then what they see happening is that you form dimers, and then trimers, and then you get a hexamer. And once you get to that point, they seem to see that the next step is to form these nanospheres in which you put several of these different types of oligomers. Once you have those groups of nanospheres, then they somehow bridge together to form chains that eventually lead to that ribbon. And since this system is now pretty well understood,
What is this day?
I'm sorry?
What is this?
Oh, that tail is this R-glu-glu-val amino acid chain, which has the charge. And so what we're doing right now, Tony is just, one of my students at Lehigh is developing a model for a spherical particle, which is hydrophobic, and then with a point charge, which we can control the charge on, to then see how this actually happens. Yeah.
The nanospheres, they're composed of not only hexamers, but also
some of these other
mixture of dimers, trimers.
Yeah, exactly. The reason I'm giving you this overview before I get to the insulin stuff, for example, just to show you how broad, widespread some of these clustering phenomena are and how different they are. And they really depend on the actual details of the forces between these things. And you see, these are orientational dependent. Again, because of the charge part. Now, this is something I just discovered a few months ago. I'm doing a lot of collaboration, I should say, with Amit Chakrabarti, who's a good friend and colleague and collaborated with Wolde, who taught me about Janus particles, which some of you may know about. In 1992, in his Nobel speech, Nobel laureate speech, Janus pointed out the potential excitement in science and technology that if you took colloidal particles, instead of having them chemically modified to be homogeneous on the surface, made them heterogeneous, if you were clever enough to do that, you could actually tune the interaction between these to create structures that you might want. And a group of chemists and physicists have now succeeded in being able to do this. So here's an example, just one of many that they've succeeded, in which you can take a sphere, a colloidal particle, and make part of it hydrophobic and the other half charged, say. And then you control the amount of charge, or I should say the Coulomb force, by putting salt into the solution and therefore screening the Coulomb interaction. And the types of clusters of super structures you get depend on the salt concentration and the nature of these forces. This just shows you this cartoon that, let's say this is hydrophobic and this is the Coulomb part, that there's a strong attractive force when the hydrophobic pieces are facing each other. As they slightly rotate, the strength of the attractive force weakens. Then you get to a point where for this orientation you have repulsive force, so you go into the repulsive region, and then you get into the extreme case with a strongly repellent. Now, if you put these together in solution and then vary the salt concentration, the types of clusters and structures you form depend on the salt concentration. So, for example, in this cartoon, this is not a cartoon, this is the experiment with different salt concentrations. And over there to the right is a Monte Carlo simulation of a model describing this. And there's almost a one-to-one correspondence between what they saw in the simulation and in the experiment. And the one I want to focus on is at highly screened concentrations, you get clusters like this. You can really create and control the type of cluster you get just by varying the salt concentration and controlling the nature of the orientational dependent forces. So this is really the way of the future, I think, in colloidal science. And what they've been able to do relatively recently is they now can control, you can put instead of a half, you can make a third, two-thirds. They really are very clever at doing the synthesis. Crystallization, since I've worked on this for a while and continue to work on it, I don't want to say too much about it, but it's really difficult to crystallize, grow good, high quality crystals. So what do? You put proteins into solution. They weakly ionize, and then there's a Coulomb repulsion between these things. And so in order to precipitate them, you typically put in a salt or work near the isoelectric point and put in something called polyethylene glycol. And then by screening the Coulomb charge, you get to a point where you can actually crystallize it. In an experiment, what you do is you have tens of thousands of little tiny samples of these systems, and you simply vary the temperature, pH, etc., concentration of protein in these things randomly until you see something grow. It's a good crystal. And that's the way it is done. Now, we understand theoretically what's going on now, but the crystal growers don't seem to give a damn. It's just reality. This is my view of how crystallographers, there's no one here, I hope. We'll come back to this, except to say one thing. There's been a recent theoretical advance that points out that until now, until very recently, people have been treating these proteins as if they're uniformly, have uniform forces, isotropic forces between them. Well, real proteins have a distribution of amino acid groups on the surface. They could be charged, they could be dipolar, they could be hydrophobic, etc. And so you really now are starting to model these with so-called patch models. And a recent PhysBio letter pointed out that those patch interactions, direction-dependent interactions, can actually aid the formation of crystals in a way that people hadn't thought about. And that's something that we're now trying to understand at a kinetic level. So let me now turn to something we've done recently, which is to talk about preparation of insulin microparticles. The standard way, apparently in the literature, to prepare drug delivery for insulin is to put insulin particles in solution and using a salt to precipitate it out. And it turns out that that works reasonably well, but that you have a problem with forming long chains. And no one quite understands why these chains result, but they affect adversely the delivery of the insulin to the patient. We read a paper that was published about 7 years ago, which took a totally novel approach that buffered the solution, worked near the isoelectric point, so you had no real Coulomb interactions. And then they put in polyethylene glycol. Polyethylene glycol is a very common thing used to precipitate because it induces an effective interaction between the proteins or colloidal particles by something called the depletion interaction. And it's understood qualitatively. And there's a well-known model, several models actually, to describe it. And the interesting thing is, for us, is that by doing this experiment, they were able to get a pretty monodisperse set of these spherical insulin microcrystals without these fibrils. And they suggested that this might be a novel way of actually preparing a system for drug delivery. So what was interesting to us is that in doing this, the first thing they actually did was to cool the system to a low temperature, and they found a fractal network of the insulin particles. They then broke up that network by vortexing it, which we think means stirring. This is the only word they have anything. And then diluting the mixture. And as I'll show you, all the evidence says this worked pretty well. So we tried to see if we could simulate that process, at least qualitatively, by assuming that the primary force here was the attractive interaction between these insulin particles that was due to this polyethylene glycol using a standard model called the Asukur-Osawa model. And I'll show you what we get in a second. So first, this is their experimental work. So they cooled the system from 77 to 25 centigrade, and then they did light scattering, continuous light scattering. After 1 second, this is a function of the scattering wave number, they didn't see very much, but after 100 seconds, they began to see the scattering intensity increase, and particularly at the smaller wave numbers. And as time went on, until 500 or so, they found that the actual slope of that scattering intensity went like q to the minus 1.8. Now that happens to be a well-known number. First of all, that tells you it's a fractal. The network is forming. And secondly, it's what you get out of a theory of, I don't know if it's theory or just simulation, diffusion limited cluster-cluster aggregation. So this was something that intrigued them. And then they did this vortexing and dilution. And then they found that the scattering intensity after that process for large Q went like Q to the minus 4. And that is, as many of you know in this room, that is due to the scattering of a compact surface known as Poirot's law. So this said that that process they went through changed the system from a fractal to a more compact behavior. Then they did this scanning electron microscopy, and this is actually the picture they got. So you can see that they actually got a reasonably uniform distribution of microcrystals, and in fact, as far as they could tell, was free of some of the problems with the other method of preparation. So this is what we were trying to see if we could at least qualitatively understand. So our starting point, which will have to be improved, but it's the simplest thing to try, is to start with the Asakura-Usama potential, which was derived in 1958. And I just learned recently that Usama was the fellow who formed the first biophysics institute in Nagoya in Japan. So he's a really very clever guy. And a relatively simple argument gives rise to this potential between two particles. And you have two control parameters. One is the volume fraction of the polymer that you put in, of the concentration. And the other is this parameter psi, which is the ratio of the radius of gyration of the polymer and the size of the, in this case, insulin particle. This controls the well depth because this function has a minimum, and that minimum is controlled by this variable. The range is controlled by this variable psi because you take the potential to be 0 beyond this ratio. The physics of this process is extremely simple. It simply has to do with the fact that if you have two large colloidal particles like insulin, say, and you put in these polymer chains, what happens is, as these colloids get together, they can squeeze out these little polymer chains. Then there are more of these polymer chains on the outsides of the colloidal particles than inside, and that exerts an osmotic pressure on these two big particles, tending to force them together, and that is the so-called depletion attraction. And we control the minimum, just to say it again, because this is important for us. We don't have temperature in this problem. This is totally temperature-independent model, and therefore the only way we can simulate the effective temperature is by controlling the well depth. If I deepen the well depth, I talk about lowering the temperature, or conversely. So we then use, that's a typo, Brownian dynamics. We started with a code that Amit Chakrabarti has been using for several years, and one of my students visited him to learn the code. We studied about 10,000 particles, and just to show you, because there's some interesting things I don't understand about this, the phase diagram for this system is moderately well known, not that accurately, but if you look at the volume fraction of the polymer versus the volume fraction of the colloid, depending on the ratio of the polymer size to the colloid size, you either have a stable phase diagram, like a stable gas liquid, then along with a solid, or, as often in the case in this field, you get a metastable fluid-fluid phase separation. And in this case that we're looking at, we have a metastable fluid-fluid. And that means there's a metastable critical point below the phase boundary. So what we did in our experiment, numerical work, is we did a deep quench back here. Now, that puts you in the region of what is called, in first order phase transition, spinodal decomposition. But this is what we saw. So you start with 10,000 particles, more or less, randomly distributed throughout the box, and then you run the code. And this is what you find after about 50 time steps. You start to see these clusters form, and as time goes on, they begin to form these more complex aggregates until you reach the last time here that I showed you. Okay? So that does not look precisely like spinodal decomposition. There's been a very interesting work going on, for those of you who follow gels and glasses, by David Waits at Harvard and others. We speculate that what you see in those cases is the extreme limit of spinel decomposition, and it looks like that's what we see here. I'm not sure theoretically how to describe that, but I'll be glad to talk about it. So then what we do is to analyze this data. I'm sorry this isn't showing better, but we plot, for example, the number of clusters that form as a function of time. And if you could see this, this would behave like 1 over time. The number of clusters decreases like 1 over the time in a wide region. We then look at the average radius of gyration in these clusters, and in that same time regime, that goes like time to 25.5. Those numbers are exactly what you get from diffusion-limited cluster aggregation models. So this tells us that we're seeing the analog of what they saw in the experiment. We then looked at the structure factors as a function of time, and this, well, how do I make this better focus?
And the floats, which are very strange.
No, maybe it's just my slides. This is deliberate, so you don't Well, what you see here in the left is a plot of the structure factor as a function of Q for different times. And it looks very much like what you saw in early stages of spinel decomposition. Peak grows and moves to the left. Over here is a plot, this is more important, where we look at the behavior of this as a function of Q, and we see that there's a region of Q to the minus 1.8, telling us that these are indeed fractals. Okay, so we get the same results out of the simulation that I showed you with the more clear light scattering slides. Now, they diluted the sample. So we just simply took the largest cluster out of this system, shown there in the left-hand top box, and they then vortexed it. We weren't exactly sure what that meant, but we decided to, in fact, heat it because we were trying to break up this network. The heat means to change the potential, the well depth, and we did that by changing the polymer concentration. So we heat the system, and then you start to see that this begins to break up into a gas of monomers and lots of semi-spherical particles, and that continues. And then we cool the system to slow down this process, and that's what you get over here. So we have several slides like this for different quenches where you end up with a distribution, of spherical clusters looking reminiscent of what I showed you in that scanning electron microscope picture of leptons.
Why is it important to remove the small clusters? The dilution, just take the largest cluster.
Well, we heat it so it breaks up.
No, first you understood it. Remove the smallest cluster. You only get.
Oh, we were just trying to. We've done it for both. We've done it for the whole system. We've done it for the largest cluster. We get the same results, but we were just trying to emulate their dilution experiment. We didn't find any difference.
You said you heat up and then you cool down, and so this means all of this is metastable, so it changes with the history.
Yeah. So yes, that's a good question. So we heat, starts to break up into monomers and spheres. That continues. Spheres grow, monomers get depleted. We then say, okay, let's slow this process down. After a while, we then get this set of spheres with very few monomers. We run that for a long time, and it stays like that. So it's a long-lived metastable state. Would it be useful for drug delivery? That's a separate.
So did you try to do the same thing instead of vortexing, heating and recooling?
I was thinking that this morning. I actually wrote, these were 3 authors at a big pharmaceutical company. I wrote each of these people, none of them replied. I then wrote to the boss of the senior person. He said, well, 2 of them quit, and the 3rd one we don't know where he is. This made me a little worried. But no, we could. I think you're correct, and I should think about that. again, we came out of this background of kinetics, the first order phase transition, so we don't vortex things. Now, another thing, we actually can do things that they weren't able to do in the experiment. We were actually able to characterize in more detail the nature of these clusters. All right. So, for example, in the case of the spheres, we can actually look at the things like the. I'll tell you what this tells us. This is a plot of, for a given atom going out and to calculating the nearest neighbor distance. You find there are about 12 atoms which are nearest neighbor. That tells you it's an FCC or HCP structure. Then there's a discontinuity to the next shell, and again, it's the second one. So we can characterize the morphology of this spherical cluster by doing that. But we also can do better by using something that David Nelson and Paul Steinhardt introduced a long time ago. You can use spherical harmonics to characterize the local order in the vicinity of a point, and then average over all points. And that allows you to see if there's any local crystal structure. And I'm really sorry this is not showing up better. But basically, what we find is that by either measure, there are two different types of bond order parameters one can use. The important point is that by either measure, we see that our spheres are made up of FCC and HCP crystals. And that's as far as we have gone. So the bottom line is we've been able to reproduce the main features of the experiment. The limitations are we could not get a reasonable solubility curve from this because our model lacks the forces that would give us temperature dependence, so that's something we could put in. More concerning to me is the question Amelia was asking. These are long-lived computer metastable states, but how long-lived and why it stays that way, I don't know. The other question I don't really understand, which is related to this gel-glass transition that is really a hot subject in the literature, if you read Nature, is can you get these fractals from a continuum theory experimental decomposition? There was an interesting paper by Bill Klein years ago who took the Allen-Kohn equation, Kohn-Hilliard, and linearized it, and then looked at what you would get from the initial exponential growth for the droplet profiles, density profiles. And in 2 dimensions, you get a fractal in this local structure. In 3 dimensions, he claims he gets a fractal, but the fractal dimension is 2, and that to me is not really a fractal. And it certainly doesn't capture what we've seen. So I don't know the answer. My friend Amit says, well, you can't coarse grain. This is happening on too fine a scale. I'm not totally convinced of that, but if you have a better idea, that would be interesting. Let me just tell you a bit about sickle cell anemia. And this is something that's been studied experimentally. I think I talked about it briefly years ago in the role of water in this. What we're trying to understand right now is what happens inside the red blood cells to the formation of the chains, polymer chains that are important for the sickle cell anemia. So a normal red blood cell, smooth, oval behavior. Sickle red blood cell is distorted, much more rigid. There's been a lot of work in trying to understand why this happens. And the key is what's inside the red blood cells are hemoglobin molecules. And if you're born, unfortunately, with the sickle hemoglobin molecule, then you can end up with the right-hand side happening to you. And that means that you have problems when the hemoglobin molecules have released oxygen to the system and they're trying to get on the way back. They get stuck if they're in that sickle state, and that's the thing that eventually deprives you of oxygen. This thing I learned very early on when I moved into this field of proteins is how small differences have huge effects. The only difference between a normal hemoglobin molecule shown here and a sickle one is that at where there is normally a glutamine, negatively charged glutamine residue, that is replaced in the sickle molecule by a hydrophobic valine. That small difference changes the interaction between the hemoglobin molecules in the red blood cell and can lead to the formation of chains and bundles. So people have been looking at this for probably 20 years in terms of nucleation theory. And the basic idea is that if I have these sickle hemoglobin molecules inside the red blood cell, there's a tendency for them to form polymer chains. by homogeneous nucleation. And once that happens, then other sickle hemoglobin molecules will adhere to the surface, and then those chains will grow into bundles by heterogeneous nucleation. But what controls the size of it and what limits these things in their radius of the bundle are things that have not been totally understood. This is a cartoon saying that the standard simple version of a bundle consists of 14 twisted chains. And twisting is due to the fact that the interaction between these sickle molecules are anisotropic. So again, it's this directional dependent interaction I was talking about in the beginning. Those anisotropies cause these molecules, when they form chains, to twist. And that's the key to understanding a lot. Now, in 2003, a good friend of mine at Drexel, who's really a world expert on this, published a physical letter with a theorist friend of his, who tried to answer what determines the bundle size. And the thing that they got correctly was that because you're twisting, these chains twist because of the anisotropy, it costs you elastic energy. And it reaches a certain point where you lose too much, cost you too much elastic energy as you add more chains that will eventually stop the process. That's the basic idea. So they did standard elastic theory and calculated the free energy. The trouble is, instead of calculating the total free energy for the forming of the bundle and minimizing that to get the equilibrium radius, they minimize the free energy density, which differs from the other one trivially by a factor of the radius squared. But you get a different result depending on this. Also, they did not take into account the fact that there was energy of interaction between chains, and that pulls them together. So we found a paper that was dealing with actin, which is the subject of one of my good friends at Lehigh, and we took their paper, which we thought really made sense from the nucleation point of view, and we redid this calculation. So the idea is very simple. These things tend not to be twisted very much. So you assume that there's a small twist. And as a consequence of the twist, you have strain energy in the plane, but you also have some bending of the chains. And you can approximate that bending for small torsion. And so you have all this elastic energy that builds up. Oops, I'm sorry. There should be an equation here. That has never stopped me before. So let me tell you what. Well, you have a term that's an elastic energy that you work out from Landau-Lifshitz, and that involves the radius of the bundle as well as the pitch, lambda. And then you have a surface energy term that it costs you energy to create a surface. So there's a surface energy term. And then there's a last term that's simply an expansion in 1 over the pitch length, lambda. And that last term, my friends at Drexel fit because it's known that from the literature there's really not just a single bundle, but there's a rather distribution of bundle sizes, and as a consequence there's a simple relationship between the pitch size in equilibrium versus the radius of the bundle. And from that they were able to determine the one phenomenological parameter they had in this model, and they got what I think is the wrong answer. So I'll skip that. We did the same thing and added to it the interaction between the fibers, which is not known, but we can estimate from the literature. And what we found was that there is actually a barrier to, so this value is R equals 0, that means a single chain. So if you have a single chain to form, and the different colors depend on the different energies you use to bind them. So, for example, if the surface tension is too large, the barrier is simply too large, and you don't form bundles. As you reduce the surface tension, however, then you find that this barrier drops and becomes surmountable, and then you find a local minimum. And that local minimum, the red is one of them, the green is another one, depending on the surface tension you put in. That local minimum tells you the bundle size, the most likely bundle size you're going to get for that given value of choice of the parameter. Way over here to the right is another minimum, which is the crystal state. These things will eventually equilibrate to crystals, but it takes weeks. And this process happens every 15 seconds. So no one worries about that. So we believe this is the correct picture, and when we calculate this minimum, given the best estimates we have for the surface tension in the literature, we find 11 nanometers, and that is indeed the size of this 14-strand bundle. So we think we have the right idea. But we're now trying to get a microscopic understanding of this. I have a very talented Chinese postdoc who seems to be afraid of nothing. That's a little scary. But he has developed a model that basically is the model people use in the modeling of DNA. And we're trying now to understand the kinetics of this process. So what we do is we have a model. So we first of all, we don't start with monomers. We start with chains of monomers. Okay? We then write down the microscopic energy for the chain. It consists of 3 pieces. One piece is just the stretching energy around the equilibrium. Another one is the bending of the chain. And the third is the twisting. And we know the elastic constants from the hemoglobin literature, so we put those in as an estimate. And then, most importantly, we put in highly directional dependent attractive interaction between chains. Then the question is, can we actually see from this model of chains and this model of these patch interactions, can we see these chains forming together, creating a bundle? Is there a nucleation barrier or not? And that depends on the choice of parameters that one puts in. And so this is just the detail, but it's a standard, it's called the worm model, I think. But they're all, I think, reasonable approximations. And then we use a simple patch model. These little colors are representing the attractive regions on each monomer that is meant to interact with another attractive region, like valine locking in with another hemoglobin molecule. Dynamically, this is not trivial, because there's now a torsion on the monomers due to the directional dependent forces, and therefore, in addition to standard Brownian dynamics of translation of the monomers, we have to write down a code that handles the rotation, and that has actually turned out not to be trivial. And so we've been bogged down for a while with that. But we've now solved that problem for this. I won't go through the details. So now we have some test results just to show you, and I will finish with this. So first we start with a single straight chain. We let it run. Now, if the model is reasonable, then it should twist and form a helix. And we find after so many steps it does indeed do that. So that's something we expect. The next thing we did is start with a straight chain and put a bent chain next to it within the range of interaction. And let that run, and we see that they start to dance together, and then up with the two both bending and forming a married couple. That looks like the correct bundle of two. And now we have found, and I don't have a picture, we put 3 on a lattice, on 3 lattice sites, and let them, and the lattice constant being within the range of attractive interaction, and they have married. So we now have 3 chains. And we then shipped this off to Pittsburgh Supercomputing Center because we've only been running with 20 monomers, and that's not going to convince anyone. And so what we hope to do is, this is now a production run mode, so we can actually, I think, answer the question of the kinetics of this formation. Now, if any of you have been listening carefully, you realize there's no barrier to nucleation here. 2 vary. And that's because we think that the parameters we put in are just such that the critical bundle size is one chain. And we know this is true from the theory that that minimum depends on the parameters of the model. So we're now tuning that so that we can make ourselves look consistent with our theory. Okay, otherwise, we prove something wrong. I think I'm going to stop here, but let me just tell you what Tony is doing, apart from the melogenin. He's taking a model, a 6-patch model for lysozyme. That means that we assume that between 2 lysozyme molecules, there are 6 regions on each lysozyme that interact, one with another patch on another molecule. And we're trying to do Brownian dynamics to understand the kinetics of crystal formation. All the work so far on this field is Monte Carlo. And Monte Carlo is not a faithful reproduction of dynamics. But we found an interesting phys letter last year for that model that worked out a phase diagram that's like most phase diagrams these days that you get from these models. I've been producing them and realizing what's the big deal? I'm not sure it means so much. All right? Because you always have these arbitrary parameters. But the phase diagram has what's called the liquidus line. And this over here is the crystal state. Now, for any of these many patch models, you get many different crystal states. And so you will have phase transitions from, say, a simple cubic to a face-centered cubic where the patches want to align. Or you'll have a phase transition from ordered FCC to a disordered FCC as the temperature comes in and makes these patches not want to align, but it's still in an FCC state. But that's not the interesting region. What people found in this Fiswell letter is that if they put the system way up here, nowhere near this metastable critical point that until now has been considered to be the controlling factor for a protein crystallization, put it way up here, they found that these anisotropic interactions that people had not taken into account in the process actually made a difference and caused the system to crystallize more easily than people thought. And this is the plot of the energy of the system versus Monte Carlo time. So here again, you start with this random distribution of these lysozyme molecules. Nothing happens for a long time. And then right up here, just before you have this sudden drop, you suddenly get a local cluster of crystals forming due to these atoms suddenly finding themselves in just the preferred orientation ordering to form a local crystal. And once that happens, then there's this sudden crystallization coming from this. It acts like a seed, nucleation seed, and you drop the energy, and then you start to form crystals throughout the system. That is a total different mechanism from what Wolde and Frenkel proposed 10 years ago that it's sort of been the standard assumption in the literature, where his idea was you work near a metastable critical point, and you take into account the fact that you have large density fluctuations, and if you have a more dense system, you can crystallize more easily out of it. And so this is a different mechanism, and we're trying to study it to see in detail how that actually happens. I'll stop here. Oh, I have to show you. I have all these friends. Amit Chakrabarti, one of his students, Sadiq, and Wei Li was a super cocky Chinese graduate student. Ya Lu is this guy doing the Brownian dynamics, and Tony. And the money, we always have to mention, as I'm sure you guys do. Thank you.
Any questions or comments?
You have just briefly commented that the question is possible to get those fractals aggregates from continuum dynamics, and you say that there is some simulations by the Cantillian equation, but that reproduce some fractals, but is linearizing it. Can you comment a little bit on that?
Well,
I am interested in this because this question I made yesterday to one student about savannas. So it's a completely trees in savannas. It's a completely different thing. But so can one get fractal structures from some continuous model?
It's a very simple argument. You simply take this nonlinear Kahn-Hilliard equation, which tells you that the initial, if you look at the Fourier concentrations of the fluctuation field, they grow like any linear theory in some region exponentially with some characteristic wave number. And this depends on the wave number and the quench. What Klein did to try to explain some earlier numerical observation by a friend of his was he simply took the Fourier transform of this and worked out what this field looks like. Okay? And it turns out, I've really forgotten what it looks like, but they're Bessel functions. And if you look at the two-dimensional version, you find that the fractal dimension is 1.5. And that's consistent with the numerical study that his friend Washington Desai had done. And then if you do the same thing in 3 dimensions, I think you get 2. But to me, that's not a real fractal. And by the way, this is just on a certain length scale. That's the only argument I know.
A combination of those Bessel functions has an empirical hypothesis.
Yeah. But I mean, it's true that nowadays simulations of microscopic models below the critical point for many models give fractals. And it never occurred to me when I first looked at the insulin paper why they were saying fractals, but given our background in continuum theories of phase transition, now I can't quite understand it. And of course, Amit would be right. This happens on such a short length scale that continuum theories don't make sense, and I don't really know.
Again, for this problem, you showed some laws of the structure factor.
Yes.
Is there anything known about the growth laws of domains, or how that would relate to this continuous model?
We're just doing that, and I don't know the answer yet. We've done some long runs and tried to analyze that, but we don't yet have results. I'm very reluctant to get back into past fields. I don't know about you, but I think that's behind me. Growth laws scare me.
You have this potential first case for the protein potential was this Japanese names. It looked to me like simple like a cubic potential, right? There was nothing else to it. So I wonder how important is the details of the potential.
Which one? I had a couple.
Or Yara. What's the visualize?
Which?
This for the assembly.
For the hemoglobin?
No.
For the Asawa. Asakura Asawa.
That's
I don't think that's
it's just a linear and cubic.
Yeah.
So
well, first of all, that's a good question because
it's very general.
That model does not work quantitatively compared with experimental. And there have been many people, so what you have to do for first principles is you start with a binary mixture of colloidal particles and polyethylene glycol. Integrate out the polyethylene glycol and find an effective Hamiltonian. And that'd be really hard.
Because it looked to me like the 5-4 model. Linear and cubic.
It's just it's just a very short range. I forget the detail, but it just gets cut off. Just a very short range attractive well. It's a single well.
There's a 5 model.
I mean, I don't think that would make much difference, but there's no first principle derivation of it, and like in most of these things, and it's a hard problem. This famous Hansen, Jean-Pierre Hansen in Cambridge has spent years trying to get a better theory, and it's really hard.
Some part of you mentioned that you were modeling the building up of these actin filaments, right? Which are twisted and so on. But if I recall correctly, actually actin filaments do not.
These aren't actin. We use a model.
Okay.
That have been.
Actin is completely different.
Actin is completely different.
Okay.
It was
Then you
proposed by.
have some enzymes. I mean, it's not self-assem.
No, it's a different problem. It's just that the elastic energy part is something that people used earlier for actin. So we just simply borrow that part. You're right.
Okay. Any other questions? No, let's Jim again.